yet, I think. Good morning or afternoon or evening, wherever you are in the world, and welcome to day 2 of the AMR Inward mission in 2001. I'm Phil Packer, and I am the Innovation Lead for AMR and Vaccines at Innovate UK, and will be chairing the session today. Just a little bit of background. This event aims to add to the global curbing of AMR. And we're going to try and do that by boosting joint, early and translational research, R&D, clinical development, validation, registration, and commercialization of vaccines, diagnostics, and antimicrobial products. And I think everybody recognizes, and we recognize that in yesterday's discussions, that we cannot do this in isolation, and we need collaborations between individuals, organizations, nationally and globally to allow this to happen. And I'm glad to say that to date, we have had 380 delegates joining this meeting for this mission. And we also have a B2Match meeting set up, which you're all aware of. And to date, we've had 160 meetings go on that. And this can carry on through the next month. So the system will be open for another month, where you can join and have meetings with your colleagues. We were hoping that Tim Jinks would be able to join us today, but unfortunately he's not been able to join us. But just before we start anyway, I'd like to do a little bit of housekeeping, and then I'm going to introduce Martin van Dugen, who we have been working with very closely in organising this meeting. So just to let you know that the session is being live streamed, and people have signed up for particular sessions. So it is crucial that we do keep on time, and we'll start the talks on the time that they're designated. And as a result, I will be butting in just to make sure all the speakers know that their time is running out if necessary. At the end of each of the talks, we have some time for questions. But if we do run out of time, there is a specific question and answer slot at 2:45 or 2:45, where we will bring all the speakers together, and we will take any questions at that point. So I also like to point out that we will be making the slides available after this meeting, and we are recording the sessions. And we also have a Twitter handle for this event, and that is at hashtag AMR Missions UK. So I'm going to pass to Martin, who will give us a little bit more information about what's going on in this meeting and some stats, I'm going to guess.
Okay, thank you very much, Phil. For introductory words. My name is Martin van Dongen. I'm a medical and molecular microbiologist. And about 5 years ago, together with 12 Dutch organizations, I took the initiative to set up AMR Insights. And AMR Insights is a globally active network-based organization, and we try to act to the extent possible to curbing of antimicrobial resistance, AMR. And we do so basically by focusing on 3 different pillars. The first pillar is that we try to inform professionals to the extent possible by means of a new newsletter, which is now sent to about 7,000 people worldwide. And we have an information platform where we welcome about 40,000 visitors per year. Our second pillar is that we also educate professionals by means of an annual AMR masterclass. I must say international masterclass, because we have contributions from many, from professionals, from experts from many different countries. And our third pillar of activities is that we also connect professionals. And we do so basically by organizing international matchmaking symposia, 2 per year, one in June focusing on the use of data technologies, and one in November, also focused on innovation and novel antimicrobials and diagnostics. And with respect to the connecting, we, together with Innovate UK, we took last year the initiative also to initiate this AMR innovation mission to the UK 2021, and also with the same aim to connect professionals with the aim to add curbing AMR, joint innovation, collaboration, which is, as we all know, very much needed to increase the curbing of AMR. Last year, AMR Insights also initiated a global network of ambassadors, the AMR Insights Ambassador Network, and we're most happy now to have over 280 of these ambassadors in some 55 countries. And you can imagine that, well, these are all very committed professionals. That these professionals join forces in order to set up projects, to realize projects. We so far also have already published several scientific publications. We will initiate a multi-country retrospective clinical study with hospitalized COVID patients where we look at AMR and AMS, and we have set up many other additional projects. So if you're interested in knowing more about these AMR insights and BEST network, please feel free to reach out and send an email or whatever. Okay, this is about AMR Insights, and coming back to this the mission, today's topic, especially the topic antimicrobials. AMR Insights is very happy to have been able to join forces with Innovate UK, Oxford Innovation, and the National Institute of Health Research to set up this very first AMR innovation mission. And in addition to what Phil already has said, the scope of this mission is very broad. I mean, we basically focus on yesterday on novel diagnostics. Today we focused on antimicrobials, but also on alternative strategies such as phage therapy. And tomorrow the focus will be on preventives and such as vaccines, but also stewardship tools. And as we all know, all these innovations, new developments, new technologies, new products, new services are desperately needed to add to the escalating antimicrobial resistance. And it's a great pleasure to work with these organizations to have this mission. And what Phil already said, we are now approaching almost 84 participants for these 3 days. That's really a great result, and it also shows how many people around the globe are really committed to the adding of the curbing of AMR. And maybe in addition to the partnering sessions, which is an important part of this mission. As Phil said, the partnering is via the B2Match platform. So far, 160 meetings have been scheduled, but also about 200 meeting invites have been sent, and these have not been answered yet. So there's an urgent request to all of you: if you have been invited for a meeting, please respond to the inviter, and we'll confirm if you're interested, or either say that you're not interested, or maybe schedule a meeting. And the time schedule for the time period for this meeting is not only yesterday, today, and tomorrow, but also for the coming 2 weeks. So there's ample time to set the meetings, and this is a very important part of this first AMR innovation mission to the UK 2021. Okay, that's what I wanted to say. Phil, back to you.
Yeah, thank you very much, Martin. I think Martin's picked up on this, but we've got over 375 people registered now, and I think we have about 63 nationalities or people from 63 different countries, and we have about a 50% academic academic business breakdown, which is really good, and I think this is really important. So, as we said yesterday, we looked at diagnostics, and I think that was a really, really good session. And I think we highlighted in that session the importance, again, of collaboration, but also the difficulties or the issues of taking something that has come out or is still in early research and actually delivering it through into a commercial project. And we had some really interesting information provided by Till Bachman on the more academic stage, and then very, very interesting talk by Mark Miller, who's talking about the commercialization, particularly of these products, and the difficulties that takes, and also 2 valleys of death that involves in getting something from the beginning to the end. And I think today we'll hear more about valleys of death from our speakers. And I think the other thing that Mark also mentioned, which was very interesting, is that possibly the problem being much larger than we thought it was. And he spoke about Lord O'Neill's report and how, in fact, that is probably represents an underestimation of the problem, because that was done on an economic analysis, but there was not enough data for all the microorganisms that are currently a problem, and therefore it underestimated the actual the problem that probably will exist in the future years to come. So that was very interesting. We also had an interesting talk from the NIH about NIHR about opportunities for people to work together with particularly the health service in the UK to try and develop their products, get them tested and get in real scenarios, and to get samples, etc., to help them do that. So all those 3 talks are available, will be available after this meeting. So please look for them online and we will send links. And we also heard from Joe, and Joe's going to talk again today about some opportunities with regard to funding in Europe. So I'm not going to steal her thunder there. And before I introduce David to speak, I'm just like to say how really Innovate, what we try to do and our purpose for our existence. So we are part of UKRI, which is the UK Research and Innovation Organisation. And we are one of the organisations with the other research councils, such as the MRC, BVSLC, which you may know about. And those other organisations mainly fund research and academic organisations, whereas Innovate UK are aiming to fund industry to try and transition things along the development pathway and through to commercialisation. So we have a slightly different reason for existence in the research councils. But this is extremely important, and it's recognised that it's very important to make the most out of the research that comes out of academia and other organisations. And that is our remit. It's basically to introduce people to each other, to organise webinars, link organisations, and to grow business by hopefully developing products. And this can be done through collaboration and also through, in some cases, direct funding of organisations. I'm not going to talk about funding today, but please keep an eye open on our website, and you can see what funding opportunities come. Sometimes they have a direct scientific area, but other times we have a continual broad call for people to put applications in. And so I'm going to introduce David in a moment, but I'd just like to say I think how it's important to have these meetings, and when we can't meet personally, I think it's not as easy. So when we first came up with this idea, well, it was probably in the margins of a meeting, and I was talking to Martin, who was helping me on something else, and we said it would be great to do something that have an inward mission into the UK. And this is a pilot, and I will say to date, it's looking really, really good. As Martin said, the number of registrations we've had and the number of networking meetings that are planned and are going on is really good. And we look forward to hopefully some really good collaborations coming out as a result of this, and hopefully applications to us in the future, but applications to other people and people building within their own organizations and companies. So I think we will start in a couple of minutes with David, and I know we've got a couple of minutes to wait. I'd just like to reiterate, so at the end of each talk, we have got plans to have a couple of minutes of questions. If we don't have time at the end of each of the talks, there is a slot at 2:45 or 2:45 where we will bring all the speakers together and we will answer questions at that point. Now, we're conscious that that is a relatively short timeline. So if we do get lots of questions, we will capture those questions and we will then get the speakers to answer them offline, and then we will put the answers on the website. Just so you're aware of that. And as we've pointed out, that the beta match session, there is a session at 3 o'clock, but also the beta match is open again tomorrow, but it is actually open for a whole month from now, so you can have your meetings in the future. So I think it's about 1:15 now, and I'm going to introduce David Powell to you, and David works is the CSO for Summit Therapeutics. He's actually a thematic champion for antimicrobials for the UK for this meeting. David is a highly experienced leader in pharmaceutical R&D with 25 years of experience in drug discovery and development across pharma and biotechnology. So I welcome David to speak, and I'll pass over to you, David.
Great, thank you very much, Phil. Hopefully everyone can hear me okay. Good day, everyone. As Phil mentioned, I'm David Powell. I'm Chief Scientific Officer at Summit Therapeutics. And it's a pleasure to be here today to tell you about the small but crucial role that biotech plays in innovating antibiotic R&D. So if we go on to the next slide, please. So the first slide here, I think, is something many of us have seen before. This charts the rise and sadly the fall of antibiotic productivity from the 1950s and 60s through to 2010s and beyond. And we've gone from a situation of having multiple novel mechanism antibiotic classes coming through from earlier periods, good commercial returns, and resistance not been a significant clinical issue simply because we had so many antibiotics coming through. However, over the last 30 years or so, that situation has changed. So we've had very few new mechanism antibiotics, and we've had incremental benefits in terms of the new drugs that have come to market. That's led to niche market positioning with poor commercial returns, and resistance now is not just a clinical, but I think as we heard from earlier on from the O'Neill report, it's also a societal issue as well. And if you click next, please. And this has also been accompanied by the real dramatic change in terms of the commercial and market for antibiotics. So branded antibiotics have dramatically decreased, and really we've now got addicted effectively from a payer point of view to generic antibiotics, which now make up the vast majority of prescriptions in this area. Next slide, please. And so the antibiotics area represents somewhat of a paradox. We know there's a high unmet need, and one can argue that unmet need is getting higher, but we have low reimbursement. And if you click again, we can actually see some of the reasons for that, which are quite complex and multifactorial. So we know that there is slow adoption of new antibiotics. So the commercials are poor, but actually to get peak sales takes quite a long time as well in this space. Resistant infection diagnosis and stratification of patient means that we have quite complex commercials and a lot of products in some areas as well. This has led to this dysfunctionality, and that has not been helped by the fact that we have few new drugs coming through, especially those antibiotics with a new mechanism of action. So how do we solve this? So on the next slide, I think I'd like to highlight ways that we can innovate, so to speak, our way out of this. And this is in a number of different areas. So on the top left-hand side, I've mentioned new mRNA antibiotics, and we'll cover some of this in a little while. Summit is also a believer in precision treatments. That is somewhat by necessity because it's actually very difficult to discover new broad-spectrum antibiotics, but also aligns with, for example, antibiotic stewardship and microbiome-sparing antibiotics, which we're also in favor of. We also believe that healthcare economics benefits should also align with clinical efficacy. And this all comes down then to kind of better clinical trial design to marry up some of these key points that I've just mentioned above. Next slide, please. So if we look at the clinical pipeline, and we're looking at a number of different things, I've highlighted 2 points here in the Pew Trust antibiotics clinical pipeline. So this is analysis of the most recent release from March of this year. And a couple of things I wanted to highlight from the recent release. So if we actually break the 40 or so antibiotics in the clinical pipeline down into, for example, where they emerge from, and we can clearly see that biotech is the real driver in terms of the clinical pipeline. And pharma represents only about 16% of the number of entries. But if we look then at innovation, and this is obviously something I think we all understand needs to improve, there's quite a low level of innovation in the clinical pipeline as well, with only about 16% meeting the WHO innovation criteria. Now I know these numbers top and bottom look similar. I just want to highlight here that it is not indeed pharma that is driving innovation. Most of the innovation is actually being driven by biotech, but the numbers actually look similar. So we have a clear lack of innovation in the pipeline all the way through, including in phase 1, for example, where only 2 of 15 entries meet the WHO innovation criteria. And the good news is, if we go on to the next slide, is that we can see more innovation in the CarbX portfolio. So if we look at the top right-hand side, once again, we see a predominance of biotech versus pharma. This might be expected. Moving to the left and looking at therapy type, we actually see that there's quite a lot of non-traditional therapies coming through now as well. They represent nearly half of the CarbX portfolio. And I think this is one of the reasons for this is because it's actually really, really difficult to discover new antibiotics, be it from small molecules or be it from natural products. And so a lot of these non-traditional approaches are coming to the fore. When we look at other aspects of innovation, we can actually see that about half of those CarbX portfolio assets belong to a new antibiotics class. But if we actually look at the new versus existing target, we see maybe a slight reduction here, and probably only about 30% of the CarbX assets come from a new antibiotic class. So moving on to the next slide, CarbX is a good segue into something I think that is absolutely key for biotech such as Summit, and that is funding. Okay, and I know there's probably going to be more talk today about push and pull funding. I just wanted to highlight really the importance of these and what they represent. So we've got the essentially the clinical pipeline from discovery all the way through to marketed product outlined below here. And we can see that push funding mechanisms are meant to incentivize the very start of the process. So these are mechanisms that provide direct funding for antibiotic research and development. Pull mechanisms are more complex. Pull mechanisms are really to sort out what we would call the dysfunctional marketplace. So they're there to reward entry to the market, to incentivize further R&D, and to ensure that patients have access to new drugs. And so there's a lot of work being considered in terms of pull mechanisms. And I'm sure that many of you have heard of the subscription models, or what people call the Netflix model, where effectively your reward for developing a new drug is decoupled from sales volume, which is very different from other therapeutic sectors. But the antibiotic sector is pretty unique when one compares it to other therapeutic areas such as oncology, for example. So both push and pull mechanisms are required. And they're critical, especially for biotech-driven antibiotic discovery and development. And the reason for that is outlined on the next slide, because our sector has got particular challenges in terms of funding for development all the way through. Okay, so I think if you click one more, we understand in other therapeutic areas there's a valley of death. Okay, so this valley of death is typically positioned around phase 1 and phase 2. This is the point at which clinical development is getting really expensive. And so you've got this valley of death until you get your proof of concept, for example, at the end of phase 2, and then very often there is an out-licensing or acquisition that brings that asset further through to the market, and we can see an upswing then, which represents obviously sales at the end of this. So this is the situation for most therapeutic areas. Antibiotics is slightly different if you go to the next click. And because essentially most biotechs really are responsible for bringing things all the way through, there's no pharma there really to pick up assets after the end of phase 2. So if you look at a lot of the phase 3 assets, these have been driven by very small companies, which is really unusual. And this means that for biotechs, for example, this valley of death is much wider and goes all the way beyond approval, for example, because you have post-approval studies as well. And just think about that, what I said about that slow tick up in cells. And this unfortunately has been a direct reason for example, some company failures. And that is because companies can get across the line, but don't really have the funding to stay in the game until profits come through sometime down the road. Typically for an antibiotics, they peak at 7 to 8 years post-launch. So the poor market returns are really driving the funding mechanism incentives that we so desperately need in this space. But the good news on the next slide is that there are some really good funders out there now. And I'm just mentioning some of the push funding here. So we've got a range of push funding that I've mentioned here in terms of Innovate UK. And these are responsible for really incentivizing this early part of the process. But you still have this gap. And if you click again and again, yeah, we know that as we actually get on towards the more expensive clinical stages, that we still have a funding gap. And this really has been an issue because there's been a limited number of players maybe such as BARDA and CARVEX, and more recently the Repair Impact Fund, who can fund in this space, but probably still not enough. But we obviously have a new entry into the field on the next click. We now have the AMR Action Fund, which is, I think, another really welcome addition to late clinical development. And they've obviously got a $1 billion fund to be able to help companies bring antibiotics to the market and through this very, very expensive clinical development phase. So that's all I wanted to mention really about, on the funding mechanism. But I wanted to mention a little bit about Summit Therapeutics' approach and how we actually feel we can contribute to the overall pipeline through our innovation and differentiation strategy. And on the next slide, we can actually see what we're trying to do. So our aim is really to differentiate. And so that is with indications of unmet need, and we're focusing on C. diff infection and carbapenem-resistant Enterobacteriaceae. We want our antibiotics to have what we call distinctive features and benefits. So we're a keen proponent of targeted antibiotics, so they only hit the infection or the pathogen of interest. This promotes good stewardship and minimizes damage to the microbiome as well, which is critical for one of our indications in particular, CDI. But then in terms of value to patients, we also want to bring in some different facets, which are more unusual, I think, in the anti-infectives and the antibiotic space, such as superiority clinical trials. Now, I know this is possible in some areas and more challenging in others, but then we also want to align our clinical trials with healthcare resource use and quality of life determination. So we end up with a much more rounded picture on what a new antibiotic represents, rather than just something which has equal or greater clinical efficacy. So on the next slide, I'm going to outline our pipeline. So these are the two projects I want to talk to you about today. The first is our phase 3 asset, which is ridinilazole, and this is targeting C. difficile infection or CDI. And the second is an earlier program, Enterobacteriaceae-driven program, which we call DDS04. And I'll be talking about a specific molecule within that series later on. Now, you'll see from the right-hand side that both of these projects have got either urgent or high threat status from CDC or WHO. And we've been very lucky. If we didn't have some of the push funders that I mentioned earlier, these projects simply would not be where they are. That's an absolute statement. So we've had funding support from BARDA, from CARVEX, from Innovate UK for the Enterobacteriaceae program, and also from the Wellcome Trust as well. And that is why these organizations are so important to biotech, to be able to advance these really desperately required antibiotics to bring these to the patient. So on the next slide, I'm going to talk a little bit about C. difficile infection. So this is, I think, a disease which is more serious than a lot of people realize. So just in the US alone, where we've got good surveillance figures, we know that there's about 500,000 cases per year. This equates to about 13,000 hospital deaths. And this is probably an underestimation. There is also a very high acute care cost associated with this of over $6 billion, and that's just in the US. And the reason that CDI is such a problem is that the initial treatment fails to prevent recurrence. And in about a quarter of cases, you get a recurrent episode of CDI. And with each episode, you get a greater increase in severity, morbidity, and mortality increases as further episodes proceed. And we understand an awful lot about this disease and actually why we get episodes of recurrence. And it's really linked to a failure of the standard of care to restrict damage on further damage on the microbiome. And so mainstay antibiotics such as metronidazole and such as vancomycin not only kill the C. difficile infection, but also take out a good chunk of the commensals as well. And that's actually what leads to further episodes of recurrence. So if we go to the next slide, please. And the reducing recurrence challenges is highlighted on this slide here. And this is really key to what we're trying to do with our new antibiotic ridinilazole. So I mentioned if you have a first infection of CDI, and this is typically after, for example, a course of antibiotics, you have then about a 25% chance of a recurrence, a second episode. However, if you then actually have a second episode or a second infection, your chances of recurrence go up to about 45% and so on. And with each episode, you're getting iller and iller, and your chance of recurrence get higher. And really, by the time you get to a third infection, this is the point where you have very, very severe inflammatory disease, toxic megacolon, and this is where gut resection and sadly, death is associated with further episodes of recurrence. And so what we're trying to do with ridinilazole is break this cycle of infection by reducing the recurrence associated with the first infection. So on the next slide, we detail the outcome of a phase 2 trial. So this was completed in 2015. So this was a study of ridinilazole going head-to-head with the standard of care vancomycin. And really what we wanted to measure here was not just cure, but a sustained clinical response. We're going to focus on the left-hand side here for a moment. And on the top left-hand side, we can actually see the comparative cure between the two treatment arms. And we can see that ridinilazole had about an 8% better cure than vancomycin. But look on the bottom left, this is really where we see the difference. So where we had quite a high level of recurrence in the vancomycin group, about 35%, we had an approximate 60% reduction with only a 14.3% recurrence rate in the ridinilazole group. And when you combine cure and recurrence to this sustained clinical response composite that you see on the right-hand side, we see a much better sustained clinical response, a big difference of over 20% in favor of ridinilazole compared to vancomycin. And the reason for that difference in sustained clinical response, we now understand, is down to the effect on the microbiome. We've done a lot of science around this, and there's more to come out this year in terms of further publications that we've been working on in terms of the phase 2. But on the next slide, you can just see a summary of this. And this is a summary of essentially the taxonomic output of fecal samples from that same phase 2 study. So the fecal samples were analyzed by a 16S sequencing to identify the taxonomy in the fecal samples in each case. And if we look at the left-hand side, we can see the effect that ridinilazole has on the gut commensals, and these are represented as this circular cladogram. Just to say that a red represents a lower abundance at end of treatment, so that's after 10 days of treatment, whereas a green represents a higher abundance at the end of treatment. So you can see that ridinilazole is pretty gentle on the microbiome. There's a slight reduction in C. difficile and other closely related Clostridioides, as might be expected, and a slight elevation of one of the other firmicutes. But look what happens on the right-hand side in terms of vancomycin, which is the standard of care. Okay, we see significant disruption in terms of the firmicutes, which are one of the key phyla within the human gut microbiota. We do obviously reduce C. difficile, but we also reduce some of the other firmicutes as well. But some of the firmicutes also go up as well as down. But look what happens if you move to the 12 to 3 o'clock section. This is represented by the Bacteroidetes and the Actinobacteria, two other important phyla associated with bile acid metabolism, for example. And these get pretty wiped down actually by vancomycin. And what happens when you change things, and other things will respond. And so we also have an elevation of the more problematic proteobacteria, and this is where the E. coli and the Klebsiellas start to bloom in the absence of some of those other species, which are eliminated by the vancomycin. So we're continuing to work on this science. It's very well-rounded now. We have a very good understanding, and we can really join up the story now between bugs and bile acids. And bile acids, I haven't had time to go into the story today, but they're a key part of this in terms of keeping C. difficile at bay in the lower GI tract in the colon. And so with this data, we've proceeded to a phase 3. And on the next slide, we can see the ongoing phase 3 design. And so we have a very similar study design to the phase 2. So we've got 2 studies ongoing, Recodify 1 and Recodify 2. We have 1,360 patients across those 2 studies. In each study, we have 340 patients being treated with ridinilazole or vancomycin, and we have a 10-day course of treatment, as outlined by the bottom. Sustained clinical response goes up to about 30 days after the end of treatment. but we're monitoring samples all the way out to and patients all the way out to day 100, measuring different endpoints as we go. So the key difference, I think, to the phase 2 is while the sustained clinical response endpoint is the same, this study is different because it is now powered for superiority. Okay, we really want to show from a superiority point of view, study design point of view, that ridinilazole is better than vancomycin in terms of sustained clinical response as the primary endpoint. A couple of other mentions in terms of some of the secondary endpoints. We want to elevate microbiome and metabolome to a secondary endpoint. And we're also looking at, as I mentioned, the health economics and to really highlight the benefits in terms of that acute costs that are associated with further episodes of CDI recurrence and infection. So this is a global study. It's ongoing at the moment across North and South America, Europe, and Asia Pacific. So yeah, keep an eye out for further announcements on this trial as they come and they're announced by the company. And so on the next slide, I just want to close out with some further details on our earlier program. Now, we haven't talked about this one an awful lot over the last year, simply because we haven't been out in the conference circuit as we might normally be, because of COVID. But this program has progressed really well over the last 12 to 18 months. So quite a different type of program. And so this is but once again, a similar type of molecule in terms of a specific approach towards an individual pathogen. So if we go on to the next slide, I'll tell you a little bit more about how it works. So just before that, we go into just the unmet need in terms of carbapenem resistance. I think we all understand that this is, depending on where you live in the world, is more or less a threat. In the US, we know that levels are pretty static, but we know that throughout the world there are significant differences depending on, for example, the different types of resistance mechanisms driven by different ambler classes, depending on where you might live in the world. So we know, for example, that NDM has challenges in India, but also in some parts of Southern Europe and in Denmark as well. And carbapenem-resistant Enterobacteriaceae are associated with much more severe illness and a much greater mortality, as high as 50% for pre-bloodstream infections. I think worryingly as well, we know that there's been obviously a lot of focus on COVID over the last year, but there is now increasing evidence of carbapenem resistance through COVID secondary infections and increased carbapenem use associated with COVID pneumonia. And that's not just carbapenem-resistant Enterobacteriaceae, but also Acinetobacter potentially as well. This is a much more challenging space from a clinical perspective than the previous example I gave, CDI. We want to attain clinical superiority where we can. I think we understand that the gram-negative space is really tough to do that. However, if you look at how you might segment carbapenem resistance in terms of different classes and different products that address that, it is a possibility, but I think it's something that obviously we will consider as we bring this molecule and this series forward. So on the next slide, I'll just detail 2 slides on one of the actual assets within this particular series. So DDSO4 we call the series. Okay, so 738, SMP 738 is one of the molecules from this series. It's a small molecule antibiotic. It was actually discovered by high throughput screening, and not that many antibiotics have come through from high throughput screening. This was discovered from a million plus high throughput screen. And it's a small molecule that targets the LAL-CE lipoprotein transport complex. And so this is an essential complex, as you can see outlined here in terms of biochemistry, that transports lipoproteins from the gram-negative inner membrane to the outer membrane. A number of different components. The LAL-CDE complex is highlighted with the square, and it is responsible. It's an ATPase-driven transporter that transports lipoproteins and shuttles them via LAL-A and LAL-B to the outer membrane. And if you disrupt that complex, it's lethal to the cell. So we started a program on this about 4 years ago. We were very lucky to attract funding from Innovate UK, and that really got that program off the ground. And this program is progressing really well. And I don't have time to talk much about it today, but I hope one slide can tell a lot. So on the next slide, we've just got the details of 738 in one of the models that we've run more recently. This is a sepsis survival model. So this is a murine model of septicemia using a beta lactamase resistant E. coli. A very severe model, must be said. And if you can actually see the vehicle, which is the dotted line on the left-hand side, mice which are infected with this E. coli succumb typically within about 6 hours. We have a control, which is TNM, which is in red. But if we actually put in a single dose, one single 20 mg per kg dose of SMT738 is enough to ensure full survival in this 4-day model of sepsis. So really encouraging results, and this molecule series is progressing really well. And so that finishes the actual slides themselves. I just want to summarize on the final slide. And so all of this is really just a summary of what I've mentioned before about how the dysfunctional market has really impacted the entire antibiotic ecosystem. And I will compare the world's response to COVID-19. It has been very dramatic, and obviously COVID-19 is much more acute. But I think COVID-19 really does show what the world can do when it comes together to fund a new infectious disease. Biotechs are now the main drivers of R&D. I hope I've shown that with the analysis that we've done from the Carbex and the Putros clinical portfolio. There is more that we can do in terms of innovation. I know it's difficult to discover new antibiotics, but as biotechs, we have to try and fly the flag for innovation and bring as many new assets through discovery and into the clinical pipeline as possible. But we need the funding to do that. I mentioned about the push and pull. They will continue to really remain important for biotechs in the years to come. And really, just the final point is around innovation and differentiation. This is really important in terms of how we address new classes of antibiotics, and because actually the antibiotics that we are using now, they will have a limited clinical life. We have to think about the future, and we have to, as an industry, ensure that we have company and pipeline sustainability that will ensure that we can bring those important new antibiotics to patients. And with that, I'll stop. I'll take any questions if we have some time. And just a final thank you to the organizers for allowing me to present today. And I'll take any questions. Thank you.
Thank you, David, very much. A very good talk. We've got a minute or so for questions. There's one I've got here, and if we get some more, we will, which will come in a minute. We'll pick up at 2:45.
Sure.
So you're doing mainly narrow spectrum therapies. So the question that's come here is, if you've got an agent which might have a broader range utility, would you advise to try and narrow to do it on one microorganism first, use that indication, and then get that license, and then broaden the indication later on?
I think that generally is how, if you look at how many companies are addressing broad spectrum antibiotics with further clinical development, you focus on one indication. You also get approval for that, and then you can actually use that antibiotic more widely if, for example, the PK and the properties of the antibiotic align with that. I think if you have a narrow spectrum antibiotic, it really is much more limiting in terms of what you can do with it. For example, a ridinilazole antibiotic is not really going to be much good against a lung infection. It's really designed to target CDI in the gut. It's gut restricted. It doesn't go anywhere else. Okay, it's really good at treating CDI. It's not going to be any good at treating anything else. This is what some people will see that as a limitation of narrow spectrum antibiotics, but it's also an advantage in terms that you can really fine tune the properties in terms of the disease that you want. But in terms of that question, Phil, yes, absolutely. You can develop a broad spectrum antibiotic against a single indication and then go more widely once you've actually shown that works.
Okay, I think there are other questions, but I will leave it there, David, because we will move on, but we'll pick those up later on if that's okay.
Okay.
Thank you very much indeed. So,
thank you. I'm
going to now introduce John Rex, who is going to talk about the future of AMR reimbursement, which is something that David has touched on, and he's going to talk about what types of products will succeed. Now, John is Chief Medical Officer at F2G Limited. He's editor-in-chief of AMR Solutions, which many of you will know. He's operating partner at Advent Life Sciences, adjunct professor of medicine at McGovern Medical School in Houston, Texas, and is pretty well known in the field. So without further ado, I'm going to introduce John. And just before I do that, I'd just like to remind people that we do have a Twitter account, which is hashtag AMR Mission UK. And without further ado, I will pass on to John. Thank you.
Great. And audio checks is working?
Yep.
Next slide, please. Well, can I do that? You can just, okay, I don't know. So executive summary. The key messages here are that reimbursement is going to change. And it's about, and hopefully we're going to drive to a place where it's societal value of new antibiotics that will matter. Not all new antibiotics will be equally valued. And you can actually today make an educated guess about the value of your project. And to give you the tools to do that, I'm going to break my talk up into a section that I call the rules of the road, the laws of gravity, and then a discussion of emerging metrics for value. Next slide. So laws of gravity, non-inferiority and superiority. Next slide. I just heard David make a comment about that. It's helpful to realize that all clinical trials reduce to one of all studies, actually, every experiment you do reduces to one of two possibilities. Either you're trying to show that something is better than something else, or you're trying to show that two things are about the same. Showing that something is better is intrinsically a lot more satisfying. It's a cleaner design. You actually, once you've done the study, you know the patients who got better, you know how much better they got. It's unambiguous. It's the preferred design. And anti-infectives were almost always forced into the other design, non-inferiority. X is about the same as Y. It's easy to do these studies badly. A lot of people are confused about them. Just the very phrase non-inferiority sounds yucky. I don't want a non-inferior drug. I want a better one. Well, the superiority is in the laboratory, and you actually don't want to see it in the clinic. At least you don't want to be the person who's in the trial where you show it in the clinic. Next slide. And that's actually the essence of it. Is that while we want these new drugs for bad bugs, when you ask for clinical data, you hit a roadblock. And think about it in terms of COVID-19. If somebody came to you and said, "Hey, I've got a new drug for COVID, and you're in the ICU with COVID, and I'd like to give you either placebo or this new drug," you say, "Well, wait, what about all the other stuff that people do? Can I have that?" You say, no, we need to do a superiority study. Well, you say, "No, you're daft. I actually want the best available therapies." And you can spin that out for anything you choose, Ebola, MRSA, COVID. Problem is nobody wants the inferior therapy or the chance of it. If there's something you think ought to work, you'd rather have that. At least it's an effort to do something. And that's the challenge with anti-infectives. We're not talking migraine here. We're talking about things that can kill you. And while lots of adverse events that can be monitored, and death's one that can be monitored, it can't be reversed. So we actually really don't like doing superiority. We actually try to avoid. We design against superiority. Next slide. And, I'm just going to say that I spend hours talking to people about this problem. And I would like, if you're really interested in it, go watch the YouTube video at the upper right-hand corner. This notion is something that you really have to let it sink in. Superiority is almost always cannot be demonstrated in your clinical studies. Next slide. You may have to hit forward twice because the YouTube video sometimes wants to play. Okay, so the standard pathway. Next slide. So instead of doing superiority directly, where I actually beat one drug beats another, what do you do? Well, this is I'm going to call it the emerging two-study path for new traditional antibiotics that have an adequate spectrum to be studied in this way. You do one standard good non-inferiority study versus a good comparator in a setting that we call the UDR, the usual drug resistance setting. And that's that term that we've introduced recently that gives you a handle for what is it when it's not MDR and it's not XDR? Is it super susceptible? No, actually there's resistance floating around, but it's the usual resistance. So it's the stuff that you can treat with, let's say, a carbapenem. And so you do one good study in that setting where both drugs are predicted to be adequate. Everybody's happy to be in your study. You do it in a major indication. You get a nice demonstration of similar efficacy in that setting when both drugs are active, and you get a nice safety database. And then in the background, you do a study where you play Go Fish for highly resistant pathogens. And you randomize versus best available therapy, if there is a best available therapy, or you do it open label if the N is too small for this. And you collect up what you can in the super resistant space. It's not wonderfully clean data. It's not all that satisfying, but it's what you can get in a reasonable period of time. And an example of this is the Plazomicin initial registration program, where they did one study in a highly resistant study versus colistin. Back at the time, that was the best drug available. And they did one study in complicated UTI. Next slide. About narrow spectrum agents, there was a discussion of that a moment ago. And my comment about narrow spectrum agents is, unless you have to, don't. This is the idea that we sometimes have called tier C. It's where the narrowness is either because the pathogen is super rare. I'm only going to study, Burkholderia, or you're looking for, I'm only going to study some super resistant thing, or it's anytime you get down to, I'm only going to study the 12. This tiny group of 12 people, you've got a real problem, because the super small trial programs just barely or not powered at all. Can you do it? Well, maybe. Do not think of it as simpler, faster, or cheaper. And it's not just a regulatory hurdle. The strength of the evidence will become frustrating. If you are tempted by this pathway, please read the papers that we've written about this. Please look at the blog notes that I've written about this. And I'll say again, if you think you want to develop a narrow spectrum agent, my first comment to you is don't. Find a way to develop it in the traditional way that I just showed you. You're much more likely to get there in non-geologic time. Narrow spectrum agents are just really, really, really hard to develop. Next slide. This is another one I get a lot of. Agents that augment. Next slide. A fair bit of interest in the early development space in things that work indirectly, and sometimes this goes under the label of non-traditional antibiotics. And I gotta say, scientifically, I love this idea. A virulence inhibitor. Why wouldn't a virulence inhibitor be an interesting thing to add to a real antibiotic and maybe improve outcomes? Why wouldn't an antitoxin be something to add to an antibiotic and improve outcomes in some way? The problem with this whole category isn't the science. I actually think the science says these things probably could be helpful. The deep problem is in the development and demonstration of efficacy. Because the antitoxin by itself, the virulence inhibitor by itself, is not going to be the only agent you give. You're actually going to want to give a real antibiotic in combination with the augmenter. And the hope is that if I give you a good therapy, or I give you a good therapy plus the augmenter, you're going to do better with the augmenter. Now, the challenge you get, first there are some technical challenges. First, your base therapy needs to work, so you can't be the case where you're developing, you actually want to do this before you've run out of good base therapies. There's a little bit of a challenge around doing PK/PD because you probably don't have an MIC for the augmenting agent, but you can probably figure that out. It's the superiority problem that kills you on this one. Next slide, let's talk about that, because it's a math problem. In your clinical trial with the augmenter, you're going to need to do existing standard of care, as I said, versus existing standard of care plus your thing, okay? And you're going to want to show that when you add it on, it's better. And that superiority has to be grounded in something the patient can feel. I got better faster. I didn't die, whatever your endpoint is. If you think about it, in most of our clinical trials, the SOC success rate is 85 to 90%. So you're already way up here in the zone in which you can get an improvement. It's about yay much. So you've got to get an improvement in that much of the overall clinical population. Some of those people just can't be saved. There's always going to be a few who will never recover. But even, but the 10% is a very tiny zone. And when you start doing the math, you discover the trials have to be huge. And the effect has got to be stonkingly strong. If I came to you and said, well, I went from 90% to 91%, you'd say, really? Do I believe that? Well, maybe you're not so interested in that. Are there settings where that kind of an increment might be possible? Well, there are a few, like endocarditis, where actually the success rates are lower than 90%. Some chronic infections, similar problem. There's more room to move. The problem is the lack of room to move. And so you've got to focus on a place where there's more blue sky above the best standard of care. This is an area where we've talked about, could other endpoints be helpful? One that comes up here is, what about a product that prevents resistance? Well, that may or may not be of interest to you personally, but it may be of interest to your community. How do you show clinical value of that? I'll say again, this is not a regulatory problem per se. The agencies are simply the first ones to point out the issue. Why should I use this drug? Which patients should get it? Why should I pay for this? So you have to think about all that if you're tempted by one of these agents. Next slide. All right, so with that as background, let's talk about the value argument and how you can think about metrics. And so we had two segments here. The first one's going to be some stories that I want you to know how to teach other people. And it's also going to lead us into some metrics. Next slide. So a question that I enjoy asking people is, have you used a fire extinguisher today? And this is one where almost everybody says, no, actually, I've been just fine all day. My house didn't catch on fire. So next slide. So then you can sharpen it up and say, hey, are you sure? Are you using a fire extinguisher right this second? And when you ask that question, people say, why do you keep bugging me about this fire extinguisher? Clearly, I'm not on fire. I'm not using a fire extinguisher myself. Well, the reason I ask this question several times is to say, well, okay, are you in a building? Are you in an apartment? What would happen if a fire broke out in your neighbor's kitchen? Would you expect something to happen there? And so the message here is that we are in buildings safely next to each other because we actually know that if something went wrong next door, the building is unlikely to burn down because we actually have a fire department. So I would argue that the only part you actually are using a fire extinguisher. The only part you're not using is the part that gets you wet. Everything else about it, you've paid for it. We are keeping it up to date all the time. There's a fire extinguisher somewhere near you, and it's got a recharge by and a recheck sticker on it, and somebody's been taking care of that fire extinguisher. It's right there available to protect you. And antibiotics are like fire extinguishers in the sense that infections go quickly, you got to have the antibiotic right away, otherwise you die. Fires go quickly, you got to have the fire extinguisher right away, otherwise you lose the building. And if you look in the upper right-hand corner of the slide, that's why the logo of the AMR Solutions website is a fire extinguisher. Next slide. And the problem with talking about this is that we're used to talking about fire extinguishers as making us safe. It's actually built into our mindset. You don't even think about it anymore, right? But with antibiotics, we're not used to thinking about the fact that antibiotics act like fire extinguishers. They are the safety net. Because you've got an antibiotic, it's safe to go get your hip. Because you've got an antibiotic, I can take care of a newborn baby who will definitely need an antibiotic at least some of the time. For lack of a good anti-COVID fire extinguisher, the world is still on fire. Another way to say it. Stated a little differently, next slide. Antibiotics, back up one. Antibiotics have values that are outside their use for individual fires. Right? And that's a key concept. For the fire extinguisher, we're used to thinking about it having for a fire, but we want it ready. What's the equivalent of that for an antibiotic? Well, we spent a lot of time thinking about that, and a very helpful acronym came out of some work that was done in the UK. And the acronym is STEADY. It stands for the 5 non-direct therapy uses or values of an antibiotic. Let me just pick out one of the 5. You can read about the others. T is my favorite to talk about because it's easy to understand. Transmission. If I have an infection with a highly resistant pathogen, I get treated, you're safe. I'm on fire, you put out the fire, can't spread to the next house. Transmission. So the existence of the antibiotic that can treat me protects everybody around me because it prevents transmission. Next slide. And it's really, I think, it takes people a while to wrap their heads around. So I've got a little story that I've been working on about COVID and fire extinguishers. And what's the value of a good fire extinguisher? Really interesting little bit of tension here. So let's all hop in a time machine. Let's go back to 2010. We've heard of SARS and MERS, and we get busy and say, we're going to invent a drug for coronaviruses, SARS, MERS, and we don't yet know about COVID-19, but our drug is going to turn out to work for COVID-19 in January of 2020. So we invent this drug for SARS and MERS, and we get it registered on 1 January 2018. And now 3 questions. What are our sales? that year? Could you have stayed in business? And what's the fix? Next slide. Well, your sales are, of course, zero. Could you have stayed in business? Of course not. There's no money coming in the door because nobody's buying your fire extinguisher. What's the fix? What's this idea of a delinked pull reward that's independent of use? In effect, somebody needs to buy my fire extinguisher because it's a fire extinguisher. Next slide. And what's the value of my fire extinguisher? This is where it gets really interesting. Let's imagine that it's now January of 2020. And we're reading about the Wuhan outbreak. And somebody calls us up and say, hey, you tell me about your thing. I say, well, here it is. And we actually get out there and actually treat some people and we put out the fire. What would our sales have been in 2020? Better than in 2018. But would they have been huge? Well, some people would have bought for stockpiling. There would have been some, but the sales would not have been wild. They would have been, something. What's the value to the global community of that fire extinguisher? Well, you can look at it by now, trillions of dollars. What's the economic loss worldwide and the people who've died? I mean, you just think of the value of that fire extinguisher had we had it 18 months ago. Amazing. Next slide. So, one of the arguments you have to make about the value of why a new fire extinguisher should have a certain value is you have to say, okay, what does it cost to invent that new fire extinguisher? And here's just some data for you. The average all-in cost to approval of a new antibiotic is about $1.3 billion. You can make it, sometimes if you're lucky, it's cheaper. So if you're unlucky, it's more expensive. There's a good summary and I've given you the quote for it that was well worked up in a JAMA paper last year. But it's not just the cost to invent it. It's also the cost to make it and keep it on the market. My rule of thumb is that it costs $25 million a year to run the plant that makes the drug. So if you've just invented it, remember it's January 2018 and we've got our new COVID drug, I still need $250 million to manufacture the tablet for the next 10 years. That's before I sell anything at all. I got to build the plant, run, and pay the people. And I've got to have $100 million for pediatric work and other sorts of stuff. So at the moment that you have a brand new molecule, you're $350 million in the hole. You've actually got to pay all this money just to keep the drug alive. So the actual all-in cost of a new molecule is something north of $1 billion, and not a bad estimate is $1.7 billion on a global basis. Let's say sometimes you might get cheaper, sometimes it might be more expensive, but that's actually not, it's a reasonable number. And usage-based income will not cover these costs because new antibiotics have typically sold less than $25 million a year. Can you do it a lot cheaper? There are people who would like to think that you can. I'm telling you that, I don't get up on any given day and try to spend more money than I have to in my development programs. We're all trying to do it as cheaply as we can. It costs a lot of money to develop a drug. And small company models are already very, very, very lean. So I don't know that on average you can make it cheaper. Next slide. So this is where, in talking to other folks, you need to explain to them why we need push and pull. And we've done a lot of public thinking on this over the past 10 years. I've listed all the various projects that have been done. And the key insight is that we need two different kinds of funding. We need push incentives that help young companies get started, things like Carbex, Novo Repair. Also push the AMR Action Fund that's about to start paying for phase 2 and phase 3 work with. It's got a $1 billion fund to deliver that. And all of this is designed to get you to the finish line. And then somebody's got to keep you going. And that's where we need these pull incentives, paid on successful approval. And there are lots and lots of papers on this. I'm going to point out there's a couple of talks to listen to about it. There's a newsletter to read about it. And the key is the next slide. I think it's the next slide, where which fire extinguishers are going to earn the reward. Hopefully, you may have to go click one more time. The YouTube thing tries to play every time you advance the slide. So the concepts around which fire extinguishers people are going to value is seen to be emerging right now. So the pull incentive, governments are not going to pay for just any old fire extinguisher. We're going to want fire extinguishers that are interesting, new, and different. And the UK is the world leader in this. They have started a subscription pilot, sometimes it's called the Netflix model, where the idea is that the UK is going to buy 2 antibiotics at 10 million pounds a year for 10 years, whether they use them or not. And if you scale that up, thinking about the UK is 3% of the G20, the UK has implicitly said that a new antibiotic has a value to the global community of $4 billion over 10 years. And the UK has laid out a point scheme that tells you how it has valued the drugs. And it's things like novelty, it's things like, spectrum of activity, it's things like bacteria covered. It's science that you can see at the moment you pick your drug. And this idea of $4 billion is actually right on target. We've done a lot of economic research that says this is the value that you need to offer. It needs to be possible to earn $4 billion in order to drive R&D. And actually, if it is possible to earn about that number, it levels the playing field. It's now equally rational to start a new cancer drug company versus a new antibiotic company. Right today, without this, starting an antibiotic company is actually daft. All you can do is lose your money. There's no way to make your money back in antibiotics right now. If we change to models like this, it will become as rational to run a little company in antibiotics as it is in anything else. And the way that we're going to engage and extend on this is by serially working on the wealthy countries to contribute their fair share. The targets have to be fair, they have to be consistently available. So far, the only sizable further effort is in the US with the thing called the Pasteur Act that we are hoping will pass later this year in the United States. Next slide. And we have to click twice. I think I've said this, the $2 to $4 billion is reward. This is what a new antibiotic really costs. 1.7 is what it costs, and 2 to 4 flattens things out. And there are the sites that tell you all the background knowledge on that. It's stuff that you need to be able to, when people ask you about this, you need to know this stuff. And that's why I give you all the detail. Next slide. And this is the message to the R&D community, but also it's the message to the payers out there. We are using the idea of saying what would earn the reward, what the point schemes are going to look like to guide R&D. And if you think of R&D as a big ship that's 10 to 15 years long, big ships turn slowly, but they do turn. And by laying out the scoring scheme, you can pick your projects to do that. And I'll let you can read the various newsletters, but go to the next slide. Let's look at the UK scheme just briefly. Here it is. This is what was used to help pick the 2 antibiotics they've chosen. And then there's still some shuffling around about precisely whether they get the full 10 million or less. I don't know the number, but it's going to be based on stuff like this. So you get points for stuff. Priority pathogen coverage, where you're talking about like the WHO priority pathogen list. Levels of unmet need, what resistance determinants you cover. What have you demonstrated that you can do with it? How novel is it? So if you think about how you're going to lay out a point scoring scheme, and the Pasteur Act says it's going to invent a point scoring scheme, there aren't very many choices. It's got to be things kind of like this. Next slide. So in summary, next slide. The AMR problem is now really well defined. After a good 10 years or more of effort, probably going back to the 2009 conference in Sweden, where for the first time we talked about incentives for antibiotics. We understand the issues, and we understand how to talk about the issues. And probably one of the most important verbal shorthands that's come out of this came out of the Drive AB project. And that shorthand is that antibiotics are the fire extinguishers of medicine. And like other infrastructure, you have to buy them in advance. They've got to be at your elbow when the fire breaks out. You can't say, oh, I'll come back and I'll see you in 10 years when I've got a new antibiotic for you. No, you need it today. And COVID's a great example of that. The possible solutions have been well studied. There aren't any perfect answers to this. We spent 3 years in the Drive AB project looking at options for this. What you need are push and pull. And push is easy to understand. The big mental shift is pull, because the idea of paying for a drug so that we have it available is so different from what we do for any other drug. But if you think about the stories I've been telling, you realize it's possible to explain that to people, and people do get it, and there's serious conversations underway about needing to figure out how to do pull incentives for antibiotics. And you've got to be doing them and make it fair and consistent, because it takes years to develop, years of efforts to find novel new agents. The reward must match the required risk. The delinked pull ties together creativity and stewardship. I didn't really talk about that, but the notion that if you're paying me to make it, but I don't have to worry about whether you use it or not, that actually feeds right into stewardship. It means put the fire extinguisher on the shelf. Don't use it unless you really have the right kind of fire. But if you need it, it's there. And the message to the R&D community is choose wisely. You want to pick a project that you think has a high likelihood of being one that people want to buy down the road. That's it. The end. Questions?
Thanks very much, John. As anybody's got any questions, please put them into your chat as usual as they come in, and I will start asking them. And again, we will have a session at 2:45 as well, where we can pick up more questions as well. So yesterday, John, we heard some suggestions that we should be having some sort of pull incentives for diagnostics as well. Which is quite interesting, and to properly do some health economics on diagnostics. Do you, slightly related to that, do you believe that there should be in the long term a policy whereby you should only give an antibiotic when there is a positive diagnosis against a particular bug? Do you think that's going to cause an economic problem for companies?
Well, I also think that one's impossible. I think that if you think about diagnostics for infections, you could apply that rule for a disease where the bacteria is never, ever normal. So take TB or gonorrhea. If I detect the DNA signature of TB or gonorrhea on you, it's not normal, ever. And that's the reason I treat you. But how about the pneumococcus, which is in the noses of a third of the people who are listening to this talk right now? Healthy people. It might be, there's a 33% chance, Phil, that it's in your nose right now. Yes. Streptococcus pneumoniae. If you, this afternoon, develop a respiratory illness syndrome, I bring you in and I detect the pneumococcus in your nose, what does it mean? It means nothing. So I actually need more information than I need to do a gram stain of your sputum and show that you actually have an excess of the pneumococcus. So the problem with the whole diagnostic area is there are times when you can't say for sure what it is. You can say what it's not. If you came in this afternoon with a pneumonia syndrome, I could tell you, well, it's not TB. But could I tell you it's not the pneumococcus? So that's a little different of a game. So I'm not sure that actually, there's a biologic speed limit to what you can do with diagnostics. It's not like on Star Trek where, you can point a thing at somebody and say, well, what you've got is, Arcturian fever. It just doesn't work that way.
Just see if there's anything else coming in. Not anything. How are we doing for time? We've got a couple of minutes. So we haven't got a couple more minutes for questions. Well, I just wait to see if anything else comes in. I just, again, remind people that the beta match session is starting at 3, but we will keep that open for a month. And we have a Twitter handle, which is, I must get that right, hashtag AMR Mission UK. I've got something else coming in now. Let me just read it before I ask you.
Question for me, if I may.
Oh yeah, go on then. Well, yeah.
How can existing early stage investors be rewarded in this Netflix model? Doesn't the pull model destroy part of the push model?
No, the existing investors, the people who want to invest early, they actually get leverage on their money. The way CarbX works, if you can scrape up 10%, 20% of the cost of your project, CarbX will give you the rest of it. So if I'm an investor, it means I put in $1, my $1 gets matched with $9 to do some interesting work. There's not a negative on that. And the idea, but another part of your question is rewarding them early. We've talked a lot about this. Should there be a stepped pull incentive that pays you out at the end of phase 1 or phase 2 or something like that? The answer to that one is no, because what you find is that anything short of approval is an endpoint that is readily gamed. You can mess with it. If you say you can earn money when you get to phase 2. By God, I'm going to declare phase 2. I mean, it's really easy. Or phase 1. I mean, it's super easy to say I made it into phase 1. I dosed one human being. Man, you've really got to make people finish the whole project. The only way we should be paying is for an approved agent. Everything else, there are too many ways for it to come apart.
Okay, just seeing if there's anything else. Martin, I know you've got would like to say something, so we'll come back to the questions later. Martin.
Thank John for a great presentation. Also a slightly confronting presentation. This mission is about joining forces, boosting collaboration. What is, from your perspective, the role of innovation and collaboration, especially international collaboration, when it comes to R&D of novel antibiotics?
Well, collaboration is helpful for the support system, having people who can do the work you need, being able to find, like the AMR accelerator is a good example of a collaborative project that provides resources to help companies get going. CarbX does that a lot. You're a little company, you know how to do this part of the program, we'll help you with the other people to do the rest of it. And I think that programs like this one, where you can find people who can help you, this is the way that we build a little virtual company. I think so. I see there's a lot of value in the ebb and the flow of the conversation, because this is how you find your partners to help you move from step 1 to step 2 to step 3 along the way.
Thank you. I think we're just about running out of time on this session. I have got another question, which I will ask at quarter 3, but I'm not sure if John can stay for that. But we will ask it there, and we'll also get John to answer it as well, because it's quite an interesting question from Alistair Parks. So at that point, I would like to very much thank John for his contribution, and I would now like to invite Jo Frost to speak. And Jo works for Innovate UK, and I will pass immediately over to her and let her introduce herself rather than me do it for her, and she'll explain what she's going to talk about. Jo, are you there? Great, thank you very much.
Yes, hi everyone, and thanks for joining us today. So as Phil said, I'm Jo and I work at UK Research and Innovation. My email address is on the slide. Next slide, please. So UKRI invest over £7 billion per year. UKRI consists of 9 parts, including Innovate UK, where I work, as well as the UK's Medical Research Council. And Innovate UK drives productivity, economic growth by supporting businesses to develop new ideas. At the bottom of the slides is a link to our UKRI Funding Opportunities webpage and funding alert sign up. Next slide, please. So today I'd actually like to talk to you about the European Research and Innovation Funding Programme, which is called Horizon Europe. And this funds people in Europe and beyond. I should say as well, my talk today is going to be the same as yesterday. So if you joined us yesterday, you can have a little break now and come back at the next presentation at past. But for those of you who didn't catch it yesterday, next slide, please. So I'm one of the UK national contact points for Horizon Europe. We're a free network of advisors to support applicants. Our network is based in the UK and around Europe and in many other countries too. Our services vary, but typically include informing and awareness raising, assisting, advising and training, and signposting. So you can contact me or the other national contact points if you have any questions. There's also a link to sign up to my newsletter at the bottom of the page. Hopefully someone will add that to the chat. And you has been funding innovation programs through its framework program since 1984. The previous program called Horizon Europe 2020 is just finished. Horizon Europe is the EU's brand new research and innovation funding program. It's just starting and will run until 2027. And it has a budget of at least 95.5 billion euros. As you can see at the bottom of the slide, the UK has agreed to associate to Horizon Europe, which means that those based in the UK will be able to take part in projects, which is excellent news. Next slide, please. So the European Commission has published a very useful Q&A confirming the UK's participation in Horizon Europe. This confirms that UK entities will have the same rights to participate as European countries. The funding rates will be the same, and also the UK can lead projects. The associated country concept is not new. The previous programme had 16 associated countries, including Israel, Switzerland, Norway and Turkey. And the exact list of Horizon Europe associated countries is to be confirmed. Other countries like Canada, Japan and Australia have expressed interest too. Horizon Europe will fund any type of organisation, including companies and universities established in EU member states or associated countries. It will also fund organisations in many low and middle income countries around the world. The exact list is to be confirmed, but in the previous programme, the EU automatically funded 122 low and middle income countries. International European research organisations are funded as well, and you can also get funding to work with organisations in other high-income countries occasionally if it's specified in the core topic or if the participation is essential for the project. So apart from that, organisations in other countries are often eligible to participate, although normally they would have to fund their own participation. And for most types of EU funding, you'll need to form a consortium including at least 3 countries. Next slide, please. On this slide, you can see an overview of Horizon Europe, which is formed of 3 pillars. I'll explain the opportunities in the various parts of the programme today, and we'll refer back to this diagram so you can navigate around. And there's some useful links to longer presentations and slide sets in the yellow box at the top right of the slide. Next slide, please. So I'm now going to talk you through some examples of Horizon Europe funding that you can get for AMR research and innovation. All the information on the slides is based on draft documents, so is subject to change, and we expect the final versions to be available within the next month. Next slide, please. So I'm going to start by showing you some of the funding opportunities within the middle pillar, circled in red. This part of Horizon Europe is split into 6 thematic areas known as clusters. For each cluster, every 2 years, the European Commission publishes a draft work programme, which contains a long list of different core topics. And for each core topic, there's typically 1 to 3 pages of information about exactly what the European Commission expects you to do in your project. So starting with the health cluster, the first bullet point in the middle. Next slide, please. So in the health cluster, there's about 50 core topics in 2021 and 2022. And the topics that relate to AMR include vaccines, developing a next generation of vaccines, and also a topic on the development, procurement, and responsible management of new antimicrobials. And the aim of that topic is to prepare for the establishment of a pull incentive for new antimicrobials, where there's an unmet public health need and market failure. So this fits together with what John Rex was talking about in his talk earlier today. I've included on the slides a link to the draft work programme and the page numbers for these topics, so you can read that to find out more. The EU also has a type of funding called partnerships. This is where the EU gets together with other funders, for example, national funders, to run joint calls. As you can see in the yellow boxes, there's 3 steps to setting up a partnership. First of all, there's some preparatory work. And then secondly, a partnership of research and innovation funders is established. And finally, as you can see in red, the partnership then holds calls for proposals. Each partnership and call involves different countries, including sometimes countries outside the EU. So you'll need to check the partnership call text to see if you're eligible for funding. As you can see at the bottom of the slide, there'll be a partnership on One Health and AMR starting in 2024. This will follow on from the current joint programme initiative on AMR, which I'll talk about later. There will also be a new partnership on pandemic preparedness. We don't yet know when this will start or which countries will be involved. And then also not on the slide, you may have heard of the Innovative Medicines Initiative, IMI. I'll add that to the slides on the website. They are also continuing and will become the Innovative Health Initiative, so you can apply to them too. Next slide, please. Cluster 2 is about culture, creativity, and inclusive society. Topics include politics and governance in a post-pandemic world, global governance for a world in transition, the role of perceptions in shaping European societies and policies. And then we've got Cluster 3, which is civil security and society, which includes topics on improved understanding of the risk exposure and public awareness in areas exposed to multi-hazards, enhanced assessment of disaster risks, adaptive capabilities and scenarios based on available historical data and projections, developing a prioritization mechanism for research programming in standardization related to natural hazards and or CBRNE threats, which is chemical, biological, radiological, nuclear and explosive threats. And the final topic in that section is improved quality assurance or quality data control for decision-making related to risk management in natural hazards and CBRNE events. Next slide, please. So Cluster 6 is, previous slide, please. Cluster 6 is on food, bioeconomy, natural resources, agriculture and environment. Topics include reaching the farm-to-fork target, research and innovation scenarios for boosting organic farming and organic aquaculture in Europe, Animal Welfare 2.0, Vaccines and Diagnostics for Priority Animal Diseases, Enhancing Biosecurity in Terrestrial Livestock Production, Ecology and Infectious Animal Diseases, and Biosecurity, Hygiene, Disease Prevention and Animal Welfare in Aquaculture. There's also some international cooperation topics, which include a One Health approach to food, nutrition, security and sustainable agriculture. Proposals for this must include African organisations. There's also a topic on agroecological approaches in African agriculture systems. Again, proposals must include African organisations. And finally, there's a topic on support for international research in infectious animal diseases. Next slide, please. So there's also a separate funding programme called the Joint Programming Initiative for AMR. And if you'd like to know a bit more about this, you can also watch Till Bachmann's presentation from yesterday. This is a global organisation, as you can see on the map. It's an international collaboration that aligns national research agendas, coordinates AMR research and funding opportunities on a global scale, and includes a One Health perspective. The JPI AMR holds regular calls for proposals, which have their own funding rules. So when future calls come out, you'll need to check which countries and types of organisations are funded. You can sometimes also get funding for countries not on the map. For more info, see the links in the grey box, including how to sign up for the newsletter. And the next call for proposals will be on AMR therapeutics. At the bottom of the slide is a summary on a recent workshop in this area, which will help scope the call. Next slide, please. So JPI AMR has a strategic research and innovation agenda, and this focuses on the areas shown on the right of the slide, including therapeutics, diagnostics, surveillance, transmission, environment, and interventions. So there's more info on the JPI AMR website. Next slide, please. So Horizon Europe also has some bottom-up funding opportunities, which are open to all research and innovation areas, including AMR. I'm now going to take you briefly through each of the programmes shown in red, starting on the left with the European Research Council. Next slide, please. So the European Research Council gives long-term funding for groundbreaking research. It funds a principal investigator known as a PI and their team. The PI can choose to do research in any field, and the projects are funded based on excellence of the PI and the excellence of the research project. The ERC has 4 main grant schemes. The starting grants are normally for PIs between 2 to 7 years after their PhD. Consolidated grants are normally for PIs 7 to 12 years after their PhD. And advanced grants are for active researchers with a track record of significant achievements in the last 10 years. Finally, synergy grants fund 2 to 4 PIs to do an ambitious project jointly that they could not do alone. Next slide, please. So this slide is about the Marie Sklodowska-Curie funding. As you can see on the top right, this funds all areas of research and innovation, and any types of organisation can get funding, including industry and academia. In general, it supports research and innovation projects that also have clear career development, as well as training and networking. The first of the schemes is the doctoral networks, which provide full funding for an international consortium to train a group of PhD students, aiming to give them experience in different sectors, for example, spending some time in both industry and academia. The second scheme is the Staff Exchange Scheme. This funds international consortiums and allows different staff working in the consortium to go on short secondments of up to 12 months per person. The third is postdoctoral fellowships, where you can get funding for someone to do a 2 to 3 year fellowship. And the final scheme is co-fund, which is where the European Commission provides extra funding for existing fellowship programmes. Next slide, please. This slide summarises the European Innovation Council. As you can see on the left of the slide, the EIC funding covers everything right from the first idea through to market. And as shown on the diagram, there are 3 main parts to the EIC: the Pathfinder, the Transition, and the Accelerator. For each of these, there's an open call where you can apply for any topic, and these calls are listed in the middle of the slide. And there are also some challenge calls where the Commission specifies call topics. These are listed on the right-hand side for 2021, and you'll have to check for future years. If you'd like any more info, then the contact points are in the grey box. The light blue box indicates the European Commission's Info Day and also their website, and you can also get the work programme which lists the call topics. And the orange box at the bottom of the slide explains that UK organisations can access the majority of the EIC apart from the EIC fund part of the EIC accelerator. So there's more detail on this on the Q&A linked to on the slide. Next slide, please. So this slide is about the Eurostars programme. Eurostars is a joint programme between the Eureka and the European Commission, and it's co-funded by national budgets from Eureka member countries. It funds transnational market-orientated collaborative R&I projects, and these must be driven by research and development performing SMEs. There are usually 2 calls a year, and we think the next one will open in July and close in October. There are 45 Eureka member countries, including the UK. These are shown in the map on the top right, and projects can be bilateral or multilateral. The rules about which countries and types of organisation are funded are explained in the call documents. At the bottom of the slide, there's a couple of other Eureka schemes, Global Stars, which funds projects including Eureka and non-Eureka countries, and each call specifies the countries involved. And there's also Network Project and Eureka clusters. I'll include some extra slides on these in the slide pack. Next slide, please. So the final scheme I want to explain is COST, which holds regular deadlines. The next one is in October. You can also get funding for any research and innovation area, including AMR. COST funds interdisciplinary research and innovation networks in Europe and beyond, for example, organising meetings, training schools, and short-term missions and networking. So COST funds the global networking of people and organisations who are already doing research and innovation. You can also join existing COST networks. For example, there's a European network on the optimisation of veterinary antimicrobial treatment, and the links are on the slide. Next slide, please. So that's the end of my presentation. I know there's been a lot of different opportunities to take in, but hopefully this overview was useful to see how you can use EU funding to work around the world, and this will save you trawling through many documents. Finally, just to let you know, we expect some information days of partnering events to be announced soon, so please do sign up to my newsletter to keep up to date. Thank you.
Thank you very much, Jo. That's very helpful. I'm not going to ask Jo any questions now because of time, but she'll be on the panel later, and as she said, you can always contact her directly. So our final presentation today is from Professor Julian Marchese from Imperial College. He's an expert in omics, and he's going to talk about the human microbiome and its defensive action against AMR. And I think this is well timed because we've mentioned the microbiome earlier on today. And this presentation will be done, and then Mark will hopefully answer questions. Sorry, Julian will hopefully answer questions at the end of his talk directly or in the session straight afterwards. So I'm going to ask if Julian's presentation can be started up, please. We haven't got any volume at the moment. Peter, can you hear me? We haven't got any volume.
System are compromised. In the context of AMR, we're interested in how the gut microbiota plays a role in providing infection control, preventing colonization by pathogenic organisms and opportunistic organisms or opportunistic pathogens, immunity, how it bolsters and trains the immune system, and how it provides support of gut barrier function, preventing organisms translocating from the lumen into the host and causing bacteremias and sepsis. The gut microbiota is a key element in preventing unwelcome bacteria. They compete for the resources that these unwelcome bacteria would require. They create a non-inclusive habitat, for example, by the production of short-chain fatty acids. However, when compromised, for example, by the regular antibiotic exposure, the conditions are then set for multi-drug resistant commensals to colonize. Moreover, I don't believe that the gut microbiota has an inherent defense against AMR. AMR is an inherent property of the gut microbiome. Resistance to aminoglycosides and beta-lactams in red here are found in babies at 6 months who have never been exposed to any antibiotics. And if you do a prospective study of individuals, for example, in China, Denmark, Europe, you find the macrolides, lincosamides, tetracyclines, streptogramin, antibiotics. The resistance mechanisms are found in all these guts. So it's a common feature of the human microbiome, or the gut microbiome. Now, over the last 5 years, we've been exploring how we can use intestinal microbiota transplant to control these MDROs in the setting of the gut. So we take healthy donors, we infuse this mixture of gut microbiota into patients and individuals with multi-drug resistant organisms to try to understand how and whether we can use this as an alternative to removing these multi-drug resistant organisms without using antibiotics. So we've been using a wide range of omic approaches on human samples, on human individuals, in mouse models. We've been using in situ and in vitro models. We've been using metagenomics or metabolite processing. We've been undertaking next generation sequencing, metataxonomics, to try to understand how an IMT, an intestinal microbiota transplant, impacts a wide range of host functions. And we're doing quite a lot of immunology now in this setting. And for us, we really broke into this area, in the whole area where people were using FMT, IMT for treating Clostridioides difficile. So in the setting of C. difficile infections, we believe a significant proportion of the way in which an IMT treats this disease is by restoring the bile metabolizing function and the ability to produce valerate. We have shown this in mouse models of C. difficile. Now, this doesn't give us an insight into how the IMT may be controlling or treating an MDRO infection or antimicrobial resistance, but we have a tool and a mechanism and a framework in which we could explore using IMT in this setting. And it all started for us when we were approached by hematologists who had a microbiological headache. They're leukemia patients who were awaiting stem cell transplant, or they had a leukemia patient who was awaiting stem cell transplant. A lot of them were heavily colonized with multi-drug resistant bacteria, E. coli and Klebsiella. And this was usually because a lot of these patients with leukemia would have many incidences of bacteremias, and they would be treated with antibiotics. They would have a fever, they would be treated with antibiotics, and they would go through numerous courses of antibiotic exposure. And eventually this would lead to selection for organisms that weren't out and out pathogens. They were commensals, but they were multi-drug resistant. They would be carbapenem resistant and a wide range of resistances. So they turned up at our door one day and said, we have this patient, we can't treat them. The last patient they gave a stem cell transplant to died of a bacteremia, which was a multi-drug resistant organism. So the patient had it in their guts. They risked giving them stem cell transplant. The patient got a bacteremia due to this drug, and they had this organism, and they had no drugs available to treat the patient. So the patient that they came to us with was in this sort of holding state. They couldn't treat them for the leukemia because of the risk of multi-drug resistant organism causing death in that patient. So they had stopped the stem cell transplant due to the risk of the the MDRO colonizing the patient and getting an untreatable MDRO and dying. So we started to explore using the IMT to modulate the gut with an aim to remove the MDRO and allow the stem cell transplant to proceed. And this was a case study. So this was a 63-year-old man presented with a new diagnosis of Philadelphia positive acute lymphoblastic leukemia. And this was the time course. So at 224 days before they received their IMT or FMT in this case, you could see that they had blood cultures, they had an E. coli in there, which was resistant to quite a lot of different organisms. Okay, minus 203 days, they still had this E. coli, then a Klebsiella came in, then they found the E. coli again. So they had this persistent colonization with a multi-drug resistant organism. For those of you who are eagle-eyed, you can also see that they were positive for C. difficile. Now, in this setting, this was our excuse, so to speak, or our in, to allow us to actually treat this patient. Because they had a C. difficile infection, we could give them an FMT, IMT under nice regulations. And so we did. But we also wanted to see whether this rescue of their gut microbiota, which had been hammered by numerous courses of antibiotics, would also have an impact on their carriage of this multi-drug resistant E. coli. So at day 0, or as we call it here, they had their intestinal microbiota transplant. At the same time, the rectal screening was carried out. They still had their E. coli. So on the same day, we showed that the E. coli was still present. On day 14, they had their stem cell transplant. And then you can see on day 16, we did a rectal swab and there was no multi-drug resistant or extensive drug resistant organism present. There were C. difficile negative as well. And this carried on. There was an E. faecalis blood culture, but it was sensitive to all the antibiotics they could be treated with. And at 36 days, they still were negative for any of the multi-drug resistant organisms that they'd been colonized with previous to having an intestinal microbiota transplant. So the neutrophil engraftment was achieved on day 25. At day 100, the patient was well, no evidence of leukemia, no graft versus host disease, or importantly, multi-drug resistant organism by rectal screening. At 12 months of post-transplant, the patient remains well and in remission. So this was a first example for us of how we could utilize IMT to treat a patient who, in all intents and purposes, was very, very ill. This must be the worst case scenario for us. This was a patient who would be undergoing an immunosuppression so they could receive a stem cell transplant. And we transplanted a whole lot of bacteria from a donor into them. This up with a case series. And what we found was that we looked at patients for multi-drug resistant bloodstream infections, total bloodstream infections, length of stay in hospital, days of carbapenem treatment. And in a small group, we also had renal patients who had recurrent UTIs, and the number of multi-drug resistant UTIs these patients also have. You can see in all these 5 outcomes here, we see a significant reduction. There's one example here of the stay going up in the one patient. The post-transplantation with a fecal microbiota, an intestinal microbiota, made a significant impact on these important parameters. Now, importantly, we didn't always decolonize the patients here, but we feel that their quality of life was dramatically improved. And so we published this about a year or so ago, and we said that disease prevention, not decolonization, is an important feature to think about here. Decolonization of a multi-drug resistant organism is a very hard endpoint to meet. And we thought that actually, if you start looking at quality of life and other endpoints and outcomes, we might actually improve the patient's quality of life by doing an IMT to treat their multi-drug resistant organism. So use an IMT to control MDROs and gut microbiome. We're now starting to explore in a much wider range of patients the potential of IMT as a rescue therapy. And what we're doing here, we're rescuing the gut microbiota that's been continuously exposed to antibiotics, such that the diversity is reduced and you're getting selection for multi-drug resistant bacteria. So we're saying, can we reset the gut microbiota to control the impact of the multi-drug resistant organism in the host? And maybe not actually decolonizing it, but reducing it down to a threshold at levels of it that actually does not become an issue for the host. Where the gut microbiota be shaped by regular antibiotic exposure and subsequent colonization by MDRO opportunistic pathogens. While I do not believe that the gut microbiota is a defense against AMR, it's a feature in everybody's gut that you will find antibiotic resistance. I think in an ecosystem which is shaped by constant antibiotic exposure, which also allows MDROs to colonize, IMT can rescue the system and modulate the levels of the MDROs such that the host is afforded some protection. And I think it's a viable alternative to using more antibiotics. So finally, I'd like to thank my colleagues and collaborators, Francis Davis, Roman Ghani, Andrew Innes, Julie MacDonald, Ben Munch, and Jiri Pavlou for all their help in this. And it's a real team effort working between basic scientists and clinicians to help bring this new and novel therapy to bear against these multi-drug resistant organisms for which we have very few, if any, antibiotics to treat them. Thank you very much.
Thanks very much, Julian. That's very interesting. And that's we have got a general question and answer in a moment, but I'm going to ask, can I ask one question now? And we might then pick it up in the next session when the other presenters are present. But I was just wondering about is there a utility of using an antibiotic which is very definitive? You may have not seen David talk earlier, but they've got one under development for C. diff. With using that with your technology as well, and what would that benefit people even more, or would you any comment on that?
I think that the idea for our IMT is to do that, sort of get it down to a drug-based approach instead of this sort of like sledgehammer to crack a nut. In our C. difficile study, we got it down to, we think, two components that are important in the whole process, which was the short-chain fatty acid valerate, which is toxic to C. diff, and the bile-modifying enzymes, which prevent germination of the spore of C. diff. So if there was an antibiotic that was specific to C. diff, that will complement it. But then I suppose you're going to have to always think about how the economic model around that antibiotics is developed to ensure that you don't get it overused and then you get resistance in the C. diff. I mean, what we believe with our method is they don't promote resistance popping up in the organism. These are metabolic toxins, or they're hitting such a fundamental aspect of its life cycle, the germination of the spore, that it's not going to actually develop resistance to the bile salt hydrolase enzyme.
Great. I think we're going to turn over now to the group question and answer. And so there'll be a moment's pause where we just do that, and then we'll restart. So if anyone's got any questions, please continue to put them in the question and answer chat, and we'll pick them up in about 20 seconds when we swap over. Okay, I think we may have swapped over there. So welcome everybody back, and we will start off some questions. I was just going to ask, before we move on, I just wondered if David got any comment from the last point we were discussing.
Yeah, I think it's really interesting when you look at this, and we're doing further metagenomics studies on our phase 2, and maybe it's something I can take offline with Julian, but that has certainly been very interesting in terms of what it's shown more widely than just the taxonomic profile. And I think I think we also need to understand our mechanism in terms of recurrence prevention is around microbiome protection. And we have a very good story, I think, as Julie mentioned, linking into bile acid mechanism. But that's one mechanism. There's going to be other mechanisms as well. Bile acids are important immunomodulators in themselves. So it is going to be complex. And I think a really interesting thing, I think, just in terms of druggability, is where some of these new other biotherapeutic products such as Ceres 109 and Finch and Rebiotics, the other companies developing these cocktails, where they fit in potentially into that sphere as well.
Okay, thank you. So I'm going to direct this question to John to start with, but please, anybody else jump in afterwards. So the question is, science of early discovery pushes use towards narrow spectrum agents, but trial difficulties make development difficult. How do we square this circle? Is John there?
I am, and the answer is you don't. I mean, it's the deal. This is why I called it the laws of gravity. I cannot fix this problem for you. If you bring me a narrow spectrum drug that only works for Klebsiella, important pathogen, I cannot conceive of a way to do a study in non-geologic time that shows just the activity of your compound for Klebsiella. And the core problem is somebody might have Klebsiella, they might have 3 other gram negatives, and would you be willing? You've got bacteria, you've got gram-negative pneumonia in the ICU, and I say, I'm going to treat you with something that only covers Klebsiella, and you say, really? Maybe I'd like to hedge my bets for a while with something else. That's the problem you've got. So if you think I wish I could make it go away, because in theory, if I had a narrow spectrum drug for Klebsiella, there'd be times when I know how to use it. I get it. I really, really understand. That's the deal. That's why I call it a law of gravity.
Has anybody else got any comments they'd like to add to that?
I'll come in there, Phil. I think if you're down at the single gram-negative organism that John mentions, yes, it is tough. I think you have to look at the indication. Okay, so we're developing 2 agents. One which is CDI. That's caused by a single bug. And actually getting back to the kind of superiority and non-inferiority. John is right. You have to have a window. If you don't have a window in terms of what you can address superiority, you haven't got a hope of making it. And resistant gram negatives are undoubtedly the most challenging area. And so we have an agent which covers Enterobacteriaceae. Okay, so it doesn't cover Pseudomonas, okay, but it does cover Klebsiella. It does cover E. coli. Okay, it covers actually all Enterobacteriaceae. It covers Yersinia, and so that is not a bug selective agent, but it's a restricted agent. Okay, and so one does have to think about how that fits into the development pathways. But there are things like LPAD, so it is a possibility. The one thing I would say is that I've done an awful lot of drug discovery. So this is right at the early phase, okay, where you're actually discovering new antibiotics, okay? And traditionally they've come from two sources, natural products and from small molecules. Small molecule drug discovery in terms of identifying new broad spectrum antibiotics has failed, okay? So people are now going back to natural products, but that is really tough because you end up rediscovering the known. So if we're not going to be able to discover new broad spectrum antibiotics, we're either restricted to the classes that we have, are agents which are narrower than the broad spectrum agents that we're currently using. Okay, so it's a bit of a rock and a hard place, and that's how we get out of it.
Thank you. We've got a question which anybody can pick up. Any possibility to reach out to the veterinary world for drug development? Does anybody like to comment back on that one?
Yeah, I'll play briefly. We spent several years thinking about the intersection between human and veterinary development as part of the U.S. Advisory Committee on Antimicrobial Resistance. And the thing that I took away from that is that drugs for the vet world are fundamentally really different in terms of what's most helpful to you. And you can do things in the vet world that you can't do in human beings. In particular, vaccines that aren't available to human beings are actually quite useful in animals because you don't need a vaccine to protect an animal for 30 years. You need to protect it for 6 weeks. If you've got a baby chick and you want to keep it from getting half a dozen different viral infections, a vaccine that is pretty good for that period of time, even if it's got side effects, is actually a very powerful tool. And the whole food industry is moving towards, I'm happy to use vaccines in my animals and better care procedures. What consumers have said they want is the label, no antibiotics. And that's actually driven a lot of very good behavior in the agricultural world. So in theory, you could work in animals with developing new drugs if you wanted to, but I don't really think that's where most of it's going. Most of it is in preventive measures, and most of that is vaccine-based.
I'd just like to say that all the slides will be available after the meeting, so that you can, and the actual presentations as well. So don't worry about that, and all the links you can then pick up there. Right, I'm just going to see there are questions. Let me just check. I've got some written here and some. So yeah. So any more questions? I'm going to ask a question which I was originally came up because I thought Tim might be able to join us today. I'm going to ask a bit of a broad question. Do you think the term AMR is hindering us in our approach to addressing AMR? I.e., do you think the general public do not understand it, and do we need to rethink how we brand it?
I'm happy to start on this because there was an interesting article, I think, in Science about language does matter. And I think it came up with the idea that a lot of people think that actually they themselves are antibiotic resistant rather than the organism. And so I think it's an interesting semantic discussion we need to have, but I don't know. I don't have an answer to it. As you can see over my shoulder here, for those who know, that is the lab in which the first antibiotics were discovered in St. Mary's College, right there, just up there, at Fleming's lab. So in the 1920s, 1930s, that's where it all started. But I don't think we've moved forward. But maybe it's a case of more about the language and how we discuss this with our patients and with the public that needs to be brought forward rather than just changing the nomenclature.
Yeah, there were some nice papers about would drug-resistant infection be a better label, DRI. I don't, and we tried to actually float the idea of DRI as opposed to AMR for the acronym, didn't stick. AMR, we're stuck with it. And so I wish there was a better label. Because it's been discussed a number of times, it's not obvious that there is one. And so you just got to start, and people actually need to engage with the science. I mean, it's just part of what you just have to teach people.
And one point that's come up, this is for anybody really, at what point should academia and business or industry or pharmaceutical company get together? So when an academic thinks they've discovered something, at what point should they approach industry? Or vice versa?
I can take that one. So I've done, in my previous role in GSK, I was in discovery partnerships with academia. And this was a model which was really, kind of as you said, it was industry looking to academia to try and identify good opportunities for drug discovery and development. It's quite common. A lot of pharma companies are doing this. I would say it's never too early to have that discussion, because you can shape your thinking. There's no point, particularly I think, in developing an asset that you think might be good without thinking about all aspects of what makes a good drug. I've spoken to a lot of academic colleagues, and obviously they focus very much on mechanisms. Once you get into drug discovery and development, you're focusing on more on properties, on PK, and so trying to bring those two schools of thought together as early as possible, I think, is very useful.
And a follow-on from me, David, I'm not asking you to tell how your company operates, but in general, do companies go and look what's out there, or do they have something in-house they've already thought of, or does it work both ways?
Well, I can only comment that when we would look, we would go to look what's out there. Okay, because I think you don't want to be, it's not really a good model to go and try to tell academics, this is what you need to do. That doesn't work. Actually, you need to actually take obviously what the academic is doing and help them to bring that on, so that that either from an early biology point of view, if it's mechanistic biology in whatever disease area you're talking about, or actually if it's in terms of a specific asset, for example, an early molecule, how you might actually want to test that in terms of proof of concept, in terms of in vivo studies, etc. It's really I suppose it's improving the quality of the output that speaks more to industry.
Okay. I have not got any more questions coming in at the moment, so just give it a couple of minutes just in case anything does come through the ether. So we are due to close at 3, but I'm sorry to reiterate things, but we have got the beta match running, and that will run for another month. So please do use that. It's a very useful tool. As we said, we've had over 180 meetings already set up, but please continue. And we do have the Twitter handle again, if you want to use that, which is hashtag AMR Mission UK. So just wait. So there's nothing else coming in a moment, but we start again tomorrow, same time at 1 o'clock, and we'll be looking at prevention and stewardship more tomorrow. And so we welcome you to join that as and when you would like. So as we've not got any other questions, does anybody
a question for me
team want to come in? Yes, Peter.
Yeah, a question for me. So this whole event is ultimately about international collaboration and encouraging people from different countries to work together to solve this problem. Do you think that the tide is in the right direction? What I mean by that is international collaboration still fashionable? Are the institutions fit for purpose? Or because you see obviously in other areas where it is going in the wrong direction. What's the panel's view on that?
It depends on how big your net is. I think Europe is doing a good job of bringing the countries together. And I was very impressed recently with the announcement of the 5-year EU pharmaceutical strategy that builds on a lot of the things we just heard about Horizon Europe, for example. But there's also the very interesting creation of, for Europe, the equivalent of what in the U.S. we call BARDA. So in the United States, back at the time of 9/11, we created this agency that sort of is the country's advanced therapeutics, advanced thing developers, not just drugs. They do lots of stuff. And we've been asking for some time, well, where's Europe part of? Well, actually, it's coming, and it's called HERA, capital letters. So it's the name of, I think, a Greek goddess. I don't really quite know the etiology of it, but it looks, there we go. We're going to try to do it, and the Europe is saying, we're going to talk about European pull incentives and get people sort of move that up to all the finance ministers and say, this is something everybody should be doing. So I think if at the right level, we are seeing some very nice collaboration. There's long been good FDA, EMA collaboration. It's a start. Rome wasn't built in a day, but we're definitely a bit further along than we were 10 years ago.
Okay, so we're approaching 3 o'clock where we're due to end. So I'd like to thank all the speakers for speaking today. Another excellent session. And if we do get any questions coming in the meantime, we will pass them on and put them on, and any answers on the website. So I will now like to conclude the event, and thank you all very much for participating, and having the rest of a nice day. Thank you very much.
Great. Thank you, everyone.
Thank you.
Bye-bye.